And I've been interested and passionate about antimicrobial resistance for some time now. My prior work involved investigating molecular mechanisms and the clinical consequences of increased resistance across gram-positives and gram-negatives. So the opportunity to do that work in antibiotic resistance on a larger scale and to be involved in the development of drugs that would be used in this patient population in the near-term future was something that was an opportunity that I basically couldn't turn down. And the opportunity to work with people like Jeff Laudit, my boss, and Mike Dudley, our chief scientific officer, was just icing on the cake for me. The project that I'm involved in right now is investigating the use of our drug Carbovance, which is meropenem plus vaborbactam, also known as RPX7009, a cyclic boronic-based inhibitor of Class A and Class C serine carbapenemases. Our study, Tango 2, investigates the use of Carbovance in patients with serious infections due to known or suspected carbapenem-resistant Enterobacter infections. We hope to have Carbovance approved or submitted for approval to the FDA by the end of the year 2016. So that is really a near-term goal. And much closer to fruition than any work that I ever did at Cornell in investigating potential novel therapies against antimicrobial resistant pathogens. The poster session that we gave was the results of our retrospective analysis of serious infections due to known carbapenem-resistant Enterobacter. And what we did was, in preparation for our phase 3 clinical trial of Carbovance in patients with known or suspected CRE infections, we went to all of the sites that are the same sites for our clinical trial and asked them to give us data on any cases of known CRE infections, complicated urinary tract infections, HAP, VAP, and bacteremia within the preceding 6 months. In doing so, we were trying to generate information on what these CRE infections looked like clinically and what the incidence was at these centers that we'd be using for a clinical trial. We knew about CRE infections in general based on the prior literature that's been out there, that patients with CRE infections have multiple comorbidities and an overall high index of severity at presentation. What we didn't realize was the extent of those comorbidities and the extent of that severity of initial illness at presentation. We also didn't realize the extent to which there is a lot of heterogeneity between infection types and between geographic regions. So our data showed that patients with CRE infections have a substantial degree of comorbidities. They have about 30% of patients have severe renal impairment, 20% requiring hemodialysis. There is a substantial proportion of patients with either hematologic or solid malignancies, and a substantial proportion of patients who are immunosuppressed. These results of knowing what these comorbidities was led us to revamp our in- and exclusionary criteria for our phase 3 trial, such that we now think that whereas our initial in- and exclusion criteria were not really overlapping the patient population at risk for CRE infections, now we are truly overlapping the target patient population for Carbovance.